Hello everyone and welcome to today's live broadcast. Vitamin D is a modifier of genomic function and phenotypic expression during pregnancy, presented by Bruce Hollis. He is a professor of Pediatrics, Biochemistry and Molecular Biology. He is also the director of Pediatric Nutritional Sciences at Medical University of South Carolina. I am Marjorie Torres of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Bruce Hollis. I will now turn the presentation over to him.
Thank you very much. The honor to speak at this. I've done it a couple other times, but the data keep getting newer and newer, so I come back to do it more again. So today I want to talk about vitamin D as a modifier of genomic function and phenotypic expression during pregnancy. During the early days of, I'll move on here. I have nothing to disclose. So during the early days, when studies were looked at for vitamin D in pregnancy, a lot of misconceptions came from those studies. In fact, there was a lot of paranoia about them, and a lot of this started after World War II and took place in England, where they were supplementing vitamin D, and they noticed what they thought was a connection between vitamin D giving during pregnancy and hypercalcemic episodes, which were basically attributed to vitamin D at the time. And then they got paranoid about giving vitamin D during the prenatal or perinatal period, so it came to an abrupt halt. The problem is what they were looking at that time was a condition called Williams syndrome, which had nothing to do with vitamin D during pregnancy. They figured this out around the mid-70s and that it wasn't a problem. But the problem that happened was, once they made this association in the 50s, and then in the 60s they started doing experimental animal work with high doses of vitamin D in rabbits. And they were inducing something that they said was aortic stenosis syndrome in these animals, and a lot of work, and it was published actually by NIH. And in 1968, a Dr. Friedman at NIH, actually, who was a pathologist, published a paper called Vitamin D as a Cause of Aortic Stenosis Syndrome, and that basically was a death knell of vitamin D use during pregnancy, and that has been carried forward even to this day. And a lot of obstetrics and gynecology people are scared to death of vitamin D because these misconceptions, unfortunately, still permeate professional organizations. This is disproven. It's not an issue, but we still have to deal with this. So the first thing that you notice about vitamin D and vitamin D is associated with, of course, is calcium metabolism and abnormal bone growth. And it was the first thing that was recognized, and oftentimes the first thing is recognized with a given causative agent is the only thing that it, from that point forth, that it does. And that's been a problem with vitamin D, is that when you talk about the things I'm going to talk about in the future, you come back to this, well, it's really not important because vitamin D we know is only a bone factor, and so it can't possibly have an effect on these other issues that you're talking about. And so if we come back to the skeletal problems even with this infant born is called craniotabes, where it's severe vitamin D deficiency in utero. And the bone, the fontanelles, you'll see, don't close at birth, and it's abnormal. Unfortunately, this isn't that rare, and as this paper shows in a Japanese study. And the good news here is that this will resolve, okay, when you initiate vitamin D supplementation in nutrition after birth. The other problems that I'm going to talk about are not so lucky, such as asthma in a newborn child, or preeclampsia or other complications of birth that they're attributed to vitamin D. And here we see the classic formation of rickets. Again, we come back to the function of vitamin D that so many people think is the only one. And you'll see on the left, the girl has vitamin D deficiency, bowed legs, rachitic rosary, because the growth plates are not functioning in this individual because of vitamin D deficiency. And when they're repleted with vitamin D, you can see that the condition is corrected and the individual looks normal and can go on, at least from a skeletal standpoint, and live a fairly normal life. So here we come back to the question is, and this was a question that's still wrestled with today, is that how do you know what a normal vitamin D level would be? And when I talk about a normal vitamin D level, I'm talking about the circulating 25-hydroxyvitamin D, because that's the compound that gives you nutritional vitamin D status. It's the compound that's measured when you go to your physician and they said, we want to measure your vitamin D content. They measure 25-hydroxyvitamin D. It has a long half-life in the blood, and it gives you the indication of how much vitamin D you have. It's not really quite that simple, but it's the best marker we have for assessing vitamin D status. And I'll go back to this really important paper in JCNM in 1971, published by Dr. Haddad and Chu, where in this paper, Dr. Haddad developed the first really good, valid method to measure vitamin D, 25-hydroxyvitamin D in individuals. And this was done in St. Louis. And they went and collected what they called normal volunteers, biliary cirrhosis patients who have fat malabsorptions, and vitamin D is a fat-soluble vitamin, so they hypothesized that it would be low in these people. And then they went and got lifeguards. Why did they get lifeguards? Well, lifeguards spend a lot of time in the sun, and as everybody knows, the sun is an integral part, sun exposure is an integral part of forming vitamin D in animals. In fact, it's really the only way that we get vitamin D, other than supplementation, because vitamin D doesn't really exist in a normal diet. We put it there in say, milk products and butter, but from a natural standpoint, it doesn't exist in our diet. We're dependent on sun exposure, and sun exposure in our society today is not used very much because of cancer, paranoia, and use of sun creams and such as that. So let's come back to the normal volunteers. So normal volunteers in this case would have been people who work in offices, physicians, whoever they could find who were, quote, normal, living the normal life, and probably didn't spend a lot of time in the sun, had a normal diet. And when they measured these individuals, the level came out to around 27 nanograms per ml. Biliary cirrhosis patients were much lower, not surprising, and the lifeguards were 2.5 to 3 times higher than the quote normal individuals. Than the normal individuals. So if they would have interpreted this data in a more robust fashion, okay, they would have determined that the lifeguards here were at the normal level. That's the level that people should have tried to attain. And there are reasons why this never happened, okay? And you go back to even today, okay, so a normal volunteer, it's not that hard to get to 27 nanograms, but if you want to get to 64 nanograms, you either do it by a lot of sun exposure or by taking substantial amounts of vitamin D supplements. And by substantial, I mean to get to an oral dose to get to where these lifeguards in would have to be a dose of between 4,000 to 6,000 IUs per day of vitamin D. Now, you go back to 1971. If somebody would have come out and said at the time, well, our daily requirement of vitamin D should be 4,000 to 6,000 units, it didn't jive with what the recommendations were, and so they believed their recommendations more than they believed the physiology, and that was part of the problem. So we went forth with normal volunteers at low levels when in fact they should have been the lifeguards, because the regulators just weren't ready to grapple with it, and even today they're really not. So, and the reason being is that people had this misconception of how toxic vitamin D was. If you come back to 1997, the first recommendations by the Institute of Medicine, they made this statement that 2,000 units of vitamin D per day was the safest level that you could take without getting hypervitaminosis D and high blood calcium. The problem was that this statement was grossly in error and was an impediment to health, to the health of human beings. And it was derived from, I can only describe as absolutely dreadful data that was published in an obscure journal from India that never should have made its way into any sort of publication, yet alone taken and recommendations made from it. And just to highlight this, 2,000 dose of vitamin D simply isn't enough to do anything. I mean, given the fact that a day in the sun from an individual, a light-skinned individual, they will make 10 to 20,000 units in that given day. So it never made sense to me how you could have 2,000 units a day be harmful, and in fact it's not. The latest UL by the Institute of Medicine, I think, was arbitrarily set at 4,000 units per day, which is better than 2,000, but where this number came from is really quite a mystery in 2010 when they set it. And we had done studies and in fact had an institutional or a drug application where we were okay to give 4,000 units in the studies I'm going to talk about. And so maybe they took it from that, I don't know. The truth is, intakes of 10,000 units a day are totally safe and used routinely, and so there's no harm in taking that much vitamin D. The 2010 Institute of Medicine position, which is the official government recommendation to do this, the Institute of Medicine reported in the 2010 report that the optimal circulating level of vitamin D was 50 nanomolar or 20 nanograms. It's the same number. If you're reporting it in nanomoles, you have to divide that number by 2.5 to get nanograms per ml. The IOM stated that a level of greater than 125 nanomoles, 50 nanograms, are potentially harmful. And this statement is especially odd, considering that humans evolved in nature for millennia where 25 level far exceeded 50 nanograms per ml. So how did the Institute of Medicine come to this? Well, the 20 nanograms was basically recommended solely from skeletal interactions. The Institute of Medicine basically at that time said, look, all we're All we believe that vitamin D has an effect on is skeleton. All these other things, cancer, pregnancy that I'm going to talk about, hasn't been proven that vitamin D is important in those things, so we're not going to make any recommendations based on observational studies, only randomized controlled trials for which there were several for bone and not many for anything else. But that second one, where a level of 50 nanograms was recommended, they broke their own rule and used some observational data that stated that levels above 50 nanograms may be inductive of prostate cancer, which again was a dreadful study. And they broke their own rules by using an observational study to make a recommendation, a harmful, potentially harmful recommendation that makes no sense from physiology. Yet they wouldn't use it to make other recommendations that have a lot more basis in science. You also have to be aware of how vitamin D is dosed can have profound outcomes on studies, how the study comes out. And by that I mean vitamin D has been dosed over many years, either by vitamin D by a daily dose, vitamin D by a weekly dose, vitamin D by a monthly dose, or every 2 months or every 3 months or every 6 months, or even one study gave it once a year. Now, common sense tells you that's probably not the best way to do things. Daily would be the best, followed by weekly, and beyond that. And again, these studies were all based on skeletal interactions and not the other things that we're going to talk about. And you have to be real careful about how you dose vitamin D in a given study. Now, just to highlight this paper was published recently by Adrian Martineau, and they were looking at how supplementation to prevent acute respiratory tract infections or how the dosing affected the results of a given study. So you can go get this paper and read it if you like, but I'm going to go to the next slide, and this is table 2 in the Martineau study. And you look at the red circle on this paper, and you read down the right side, it says dosing regime, okay? The first one, it says bolus dose of greater than 3,000 IUs given. Bolus dose means it was given in one single dose in a week or a month or whatever they did to mimic, how much they got in a given time. There were 10 trials that did that, and then under that it says bolus dose not giving, which would mean a daily dose of vitamin D. And then how did that affect study outcomes? If you look under the p-value, you can see if on infections, acute respiratory infections, if it was given in a bolus dose, it failed to be effective. P-value of 0.67. However, if it was given as a daily dose, and I'm sorry, but that number's covered up, it was significant at a very high level, meaning that if it was given on a daily dose, it was effective for preventing infections where a bolus dose were not. So if you go beyond skeleton and dosing, I think that you have to be really careful about how you give vitamin D. And the reason that they would give bolus dosing is for convenience. They would feel, study people would feel that it would be more compliant if people took it once a week or once a month as opposed to every day. But when it's going to infect your study outcomes, you really have to. And so this has been one of the major confusions on all these studies published. That vitamin D works for this, it doesn't work for this. A lot of it has to come down to how it was dosed in a given study. And this slide is complicated, and you can go see this slide in where it was published in Journal of Clinical Endocrinology and Metabolism by myself and Carol Wagner a few years back, and it's explained better in that paper than I can do here. I could spend the whole time just explaining this figure, but you have vitamin D enter in either from diet or sun exposure on the left there. The half time of vitamin D in the circulation is very quick. It comes and goes very quickly. if you look right below it, you'll see the endocrine portion of the vitamin D system, which everybody knows, and that's what we've been taught for decades. And it's the one that functions through skeletal integrity, where vitamin D is metabolized to 25D, and then it's metabolized in the kidney to the active hormonal form of 125, and then it goes about, it's put back into the circulation from the kidney and then acts as an endocrine portion primarily to handle skeletal health. The one that is not talked about and functions as equally important and is coming to light would be a paracrine endocrine function, and that would be on the right in that orange circle, where vitamin D itself would diffuse into given tissues, breast, colon, whatever, okay, and it would go into those tissues where within those cells it can be activated by the enzymes present to the active form of 125, and then go about its function of controlling gene suppression, activation, whatever. And it's known that vitamin D, first of all, the receptors for the hormonal form of vitamin D, the 125, are located almost in every tissue in the body. And also in a lot of those tissues have the ability to activate vitamin D in a paracrine or endocrine form to use them within a given cell to control gene function. How many genes are there that could be affected by vitamin D? Well, give or take about 10% of all our genes, around 2,000 genes, have vitamin D response elements in the control region that somehow respond to vitamin D in a positive or negative fashion. So it's vast. And very few of those genes have anything to do with calcium homeostasis. So now we move into the public health and the real meat of this presentation about public health issue and deficiency of vitamin D during pregnancy. We first got interested in it. I've been interested in it for decades, but you really couldn't do any meaningful work until about the year 2000, because you couldn't give meaningful amounts of vitamin D to a pregnant woman or pretty much any other person when the Institute of Medicine said that 2,000 units or more was harmful. You couldn't get these studies through an institutional review board or an NIH study section, because they would fall back and say, well, you can't do this because you're going to hurt the patient, when in fact those recommendations weren't even enough, okay? And so we instituted, Dr. Wagner and I instituted these studies in 2000, wrote a grant, sent it to NIH. We wanted to determine what the true requirement of vitamin D in pregnancy is based on skeletal function, and also how much vitamin D it would take to normalize 25-hydroxy vitamin D in a pregnant woman. We said a normal level should be 30 nanograms or 75 nanomoles per ml. How much would it take to get them there? So we went to the drawing board, we wrote up a grant, sent it to NIH, and we wanted to give up to 4,000 units of vitamin D per day to these women. Now this is a decade or more ago, okay, even 15 years ago. So the NIH thought it was a great study, the study sections thought it was a great study, but when it came back to, and they funded the project, when it came back to our institution, predictably, it went to the Institutional Review Board, and they said, wait a minute, you have to, we will not let you do this study because you're giving 10 times the amount that the professional societies say is necessary, and we just can't allow this. You have to go to the Food and Drug Administration to get their permission to do this. And so we sent an application to the Food and Drug Administration, which is called a non-exempt IND, which is a couple of pages, and you fill out, and then they say, yes, go ahead with the study, which we anticipated, but what they said was no. They said, you have to write a full investigational drug application before we will allow you to do this study. And what that meant was we spent the next 6 months writing up, this is what drug companies have to do drug studies, writing up an investigational drug application with vitamin D proving to the FDA that we were not going to hurt anybody. And so we wrote it up, and lo and behold, about 6 to 8 months later, FDA awarded us an IND. You can see at the bottom of this thing, it was the first ever awarded to an investigation for a vitamin. You know, they do this for drugs. That's how paranoid they were about assessing or giving a study. Anyway, the study went forth. It was a randomized controlled double-blind placebo study. Basically, women, it's approximately 16 weeks when they came in to discover they were pregnant, were randomized and put on the study. In this first study, there were all sorts of safety recommendations. I mean, it was being monitored by the NIH. It was being monitored by our Institutional Review Board. It was being monitored by the Food and Drug Administration for any adverse events that we would see in any of these women. So it was very expensive and very time-consuming. You can look, this paper was published in 2011 in the Journal of Bone and Mineral Research. It's been cited 500 times. It's one of the most cited papers during the time now that in this period it continues to be a landmark paper, and if you want to go and read it, it's very long and it's very detailed, and you can read everything that we did in that study. And basically, it was also beyond that, it was a safety study. Did anybody get hurt from doing this? The short answer is we saw not a single adverse event in any of these women due to vitamin D supplementation. When we looked at what was required to normalize vitamin D to the level that we wanted, the groups were 400 units, 2,000 units, or 4,000 units per day. It took 7 years to complete this study. When we looked at it, we saw no adverse event due to vitamin D supplementation. It was quite unremarkable for anything skeletal-wise, because basically in this population, these women were really not vitamin D deficient enough to see any. And unlike, if you look at what the level should be, the skeletal system and the endocrine system requires not as much vitamin D as these other things like infection. They're two separate systems. One's endocrine, one's paracrine and endocrine. So when you're talking about what the normal level should be, you really need the skeleton, not much, and other things, the prevention of the things that I'm going to talk about, preeclampsia, asthma in children. That's a different story. You need more. But when we finally finished this study and we broke down data, we collected a lot of data. Okay, you have to keep in mind that the things that I'm going to show you now were secondary analysis, because in 2000 we had no idea that vitamin D would function beyond in any of these things. So when we looked at in an intent to treat here on secondary analysis, lo and behold, if you look at the blue numbers on there, we found what they had been finding in some observational studies. We found decrease in cesarean sections, significant decrease due to vitamin D supplementation and complications of pregnancy, comorbidities, and you can see now how is this received in the community at large. And basically, I will tell you that they thought we were insane. This couldn't possibly be true. It was presented at a meeting, and it was really a lively conversation with the people that were there, classic skeletal people. When they saw this data, they just would not believe it. So, how effective is vitamin D at reducing say, complications of pregnancy? Well, in the study that I showed you that we didn't have really severe vitamin D deficiency. And so the studies were interesting but not remarkable. This study that I'm showing you from this graph was published in Clinical Endocrinology a couple of years ago. It was done in India, where we couldn't do this study in the U.S. because these patients coming in the first trimester were profoundly vitamin D deficient, and they were left like that. In our country, if a woman now comes in a study and they are profoundly deficient, you can't give them nothing. You have to give them standard of care, which is 400 to 600 units a day by the governing organization. So they get something. In India, they didn't have to give them anything. So what they did was they had a placebo group that got nothing, and they were profoundly vitamin D deficient. And then they had vitamin D supplemented groups where they gave a substantial amount of vitamin D, and they monitored complications of pregnancy. And when they supplemented these women, they found a profound decrease in complications of pregnancy. It was astounding. Look at that graph. So there was no question that vitamin D would have, this is extraskeletal, this preterm labor, gestational diabetes, and all the things that go along with complications of pregnancy. So it was clear that vitamin D was associated with these things. So let's talk about preterm birth. All right, it's one of the things that we've looked at now, and over the last several years, once we determined that vitamin D had these effects beyond skeleton. So this study, we've looked at several different studies now. We've looked at the original NIH study. We had another study called with the Thrasher group. Thrasher funded some of our pregnancy research. The Kellogg Foundation funded research, and we've looked at this in several publications now, several studies. And what this graph shows is, by vitamin D status, how much preterm birth is associated with circulating 25 when these children are born, and the mother's levels. And you look at this graph, and it doesn't look very remarkable, but you see that gestational age goes up as 25 hydroxy levels are higher in the pregnant women. So let's expand this to a different, highlight the area, and you can see that the gestational age increases as these women go up, to the point where about 40 nanograms per ml seems the spot where you at least want to be during pregnancy to prevent complications of pregnancy or preterm birth. And that's what we saw from our early data. 40 nanograms, give or take, is really the level that you want to be at during pregnancy. Now these were done during pregnancy, not when somebody was pregnant or got pregnant, conception. And here's another view of that graph. Again, these studies are published by myself and Dr. Wagner and other investigators. You can go on PubMed, you can find these publications, and you can read them. So this is the hazard ratio. So as your vitamin D level increases, your hazard of preterm birth goes down. So, of course, when you're doing research like that, other people take notice of it, and that we had been given an institutional IND to study vitamin D. We were approached by a lot of people to do other studies. And I think the most interesting one of all was done when the Harvard group of Scott Weiss and Gus Latonia contacted us in 2007 with this idea that vitamin D supplementation during pregnancy might decrease the asthma rates in children after birth, meaning you give vitamin D to the pregnant woman, there's going to be changes initiated in utero that will help children not get asthma after they're born. Okay, so the VDART study was born, vitamin D antenatal asthma reduction trial. It was a huge study. It was done at 3 sites, clinical sites, one in Boston, one in St. Louis, one in San Diego. It was a huge study. It cost millions of dollars to do. It's still ongoing today, and there's been an incredible amount of data that was generated from this. And so we were part of this and still remain part of this today. So what have we found, and Weiss and Latonia here found from the VDART trial? Well, the hypothesis was vitamin D supplementation to mothers during pregnancy put children at risk for asthma and other allergies. That was the hypothesis. And here's the VDART study design. You can go find this. It was published in the reference there, dictating trials on clinicaltrials.gov. It involved basically giving women in the treatment arm 4,000 units of vitamin D a day, and in the placebo arm, women got 400 units of vitamin D a day. There were nearly 900 women at 3 sites in this study, and you can see all the things that were looked at on this study. But again, it was asthma was the primary one. And so how did this come out? It was ultimately published in JAMA. And the results we thought were striking, but if you come back to how, again, the standard medical profession views this, is it doesn't jive with Institute of Medicine. And to me, is that if you come up with positive results, it's not very well liked. Okay, they just don't want to see it. And so when this data was analyzed, it was analyzed in an intent to treat. There's always problems with intent to treat. There's a lot of non-compliance of the subjects, and no different in this particular group. And in one subset of these individuals, we know we had greater than 50% non-compliance, which means those people would be considered treated but were actually placebo. And when you come right down to it, given those numbers, it's ironic, it's incredible you see any effect whatsoever. However, if you looked at this data, how the treatment affected the 25D levels in the mothers given 400 or 4,000, you can see at enrollment, the vitamin D levels were 50 nanomoles or so. And then at the 3rd trimester of treatment, either 400 or 4,000, it was significantly higher in the mothers given the 4,000, although it's approaching 40. The mean is approaching about 40 nanograms or so, is where you want to be. So it's not like women getting 10 times of vitamin D have 10 times the circulating 25D. It just doesn't work that way. And then if you looked at 1 year after delivery, the levels are pretty much the same in the mothers. Now keep in mind that the vitamin D was only given during the pregnancy period. After delivery, no more treatments were given, and the patients were followed. And these are the levels in the infants. In the cord blood, again, you can see that the cord blood from the mothers who had taken 4,000 units were higher than the 400 units. It's not startlingly high, it's just significantly higher. And then at year 1 and year 3, they're equal, which you would expect because there's no further vitamin D treatment after pregnancy and after birth. So what do we find in this study? Okay, what they found was you can see this graph of the different rates of reduction, but basically what we found is the development of asthma was lower in the infants the mothers got 4,000 units versus 400. Now this was assessed in the infants at 1, 2, and 3 years. Okay, at 3 years, by intent to treat, the significance was 0.051. At the other points, it was better than 0.05 or better. To publish this paper, the deal was in JAMA that we had to state that those were non-significant studies because it was 0.051 and not 0.050. The authors made a decision to go with it, hoping that the public would see and make their own decisions, but it was put into the news as vitamin D has no effect, a non-significant effect on asthma generation, which was simply not true. So in the JAMA paper at year 3, vitamin D was demonstrated to prevent, again, at 0.051. If it would have been 0.050 or 0.049, it would have had to say it was significant, but JAMA demanded the author state that this study was therefore non-significant. However, okay, this is a straight intent to treat data, and you have to, that's what drug trials use, and there's all sorts of problems with that doing nutritional studies like vitamin D, because there's compounding factors. Everybody has some vitamin D in their blood. How does that affect the final results? So if you take baseline levels of the mother into account, a very different picture emerges. And what do I mean at baseline levels? I mean the levels that were those mothers had in their circulation when they entered the study, probably owning to the fact is what those levels were at conception, because they weren't taking any vitamin D. We hadn't given them anything yet. And these were lifestyles, okay, of how people's vitamin D's, be it sun, were they taking supplements, but what they were doing when they got. So how did that affect the outcome when we looked at mother's first trimester vitamin D levels? Profound, okay. Here's the effect in supplementation and the odds ratios of the children developing asthma. If they entered into the study, the women entered into the study with vitamin D levels of, these are at the bottom, these are levels in the first trimester of what these women were when they came into the trials, less than 20, 20 to 30, 30 to 40, and greater than 40 nanograms per ml. If they entered in with those levels and were put on placebo treatment, there was maybe some protection. It wasn't very startling, okay? However, if they came into the trial with, in the green here, with these levels, okay, 20, if they came in at less than 20 nanograms and were put on 4,000 units, not much protection was offered. 20 to 30, more protection, 30 to 40, more protection, and greater than 40, basically, in this study, eliminated the asthma in the children at 3 years. Now, we weren't allowed to present that data in the original publication because it violated intent to treat, but it was subsequently published in with the group in JACI, Journal of Clinical Allergy and Immunology, which is a very respected journal, and the results are totally different when you look at it this way. And what does this mean? It means that you have to look at what the vitamin D levels are at conception. And it's not as simple as just giving vitamin D supplementation during pregnancy. If you would look at this trial and the results aren't very astounding. It's more complicated. And what this means is there's development going on right after conception and probably in lung development that can't be reversed later by vitamin D supplementation in these studies. And ironically, there was another RCT going on besides the first one here. There was another one going on, one Lutonian, and there was another one going on in the COPSAC study. They were published at the same time. They both just missed significance. They were similar types of studies. They were both published at the same time. And what was done later is the groups got together and reanalyzed that data, putting the data all together to increase the sample size. That paper is just going to appear in Press and PLOS One, and when you do that, you come up with highly significant, even intent to treat studies. So vitamin D supplementation during pregnancy can prevent asthma, development of asthma and wheeze in the child at 3 years, and we're looking at 6-year data. And again, when we presented this data, the combined analysis to JAMA, they didn't even review it. They didn't want to hear about it. So it doesn't end up in JAMA, it doesn't fit their narrative, and so that's where you go. Finally, I want to move around here. Secondary analysis of VDART, we want to look at preeclampsia and preterm birth based on the factors that we saw. How does vitamin D affect preeclampsia and their preterm birth? And if you, this was published in Journal of Clinical Investigation within the last year or two, and you can search that out on PubMed. And if you look at this again, how does it affect preeclampsia? Well, it's similar to asthma, okay? And if you look at the paper, basically what low means, low, high, low, and high. Low means they came into pregnancy with less than 20 nanograms in the first trimester, say at conception, and they were put on the low dose of vitamin D, 400 units during pregnancy, and that was the odds ratio. That was our baseline level. If they came in low at 20 and were put on the high dose, they got some protection against preeclampsia. If they came in high and were put on the low dose, they still had significant protection, meaning that whatever was happening in preeclampsia, again, a lot of it was happening at conception or in the first trimester, which, of course, in our studies we really missed the first trimester. And then if they put them on high, meaning they came in at 40 nanograms and they went on the high dose, there was an incredible protection against preeclampsia in these women. Basically, it was eliminated. Now let's go to this slide that was published in the JCI paper, and it was the risk for preeclampsia based on the bottom axis of what the vitamin 25D levels were at conception, basically first trimester. And if you look, what you won't see in the figure in the journal, in this figure is you won't see the cases and controls. They wouldn't allow that to be published. And basically what that shows is the cases of preeclampsia at the top versus the controls at the bottom. If vitamin D at 40 nanograms, okay, if you look at that, offered perfect protection against preeclampsia, meaning that if a woman came into pregnancy with 40 nanograms or higher, they were completely protected against preeclampsia. There's not one case out there, and by statistics alone, there should have been 6 or 7 cases out there based on how many people were out there, and there were none. So, and this is a very important study, is that preeclampsia is highly linked to early vitamin D status in mothers. And you can, again, here was how the study was conducted. There was a lot of genetic signatures in here. It was in the JCI paper. You can go and read it. It's just too involved to look at here. And these are the 10 identified gene sharing networks that we found in the analysis of the JCI, basically involving gene clusters that are in autoimmune disease, inflammation, in infection. These are the things that vitamin D affects if you look at gene clusters on basically any disease you look at. Preeclampsia, cancer, they're basically affecting the same gene profiles. So let's go to the next slide. Again, you can see this cobweb of gene interactions from the preeclampsia that were discussed and plotted by gene array analysis in our preeclampsia study, and you can see the gene clusters that are affected between our placebo, which is 400 units, versus our treatment, which is 4,000. So these are the genes that are being altered by the vitamin D supplementation during the pregnancy period, totally due to vitamin D supplementation. So the conclusions. Mothers taking the current recommended level of 400 IUs of vitamin D per day will not provide adequate vitamin D. That's a different, actually a different paper. Vitamin D intakes of 10,000 are normal for treatment of physiology. Vitamin D is essential for pregnancy, not only for skeletal integrity, but also for prevention of birth complications such as preeclampsia, development of childhood asthma, and scarier things like probably neurodevelopment, development of autoimmune diseases such as type 1 diabetes and multiple sclerosis. Design of vitamin D dosing regimes for clinical trials based on normal physiology and not convenience and compliance. And what that mean is they should be giving at a reasonable dosing regime, like daily, no more in pregnancy, nothing beyond weekly doses. And the levels given should be based on normal physiology, meaningful levels of 4,000 to 6,000 units and not 400. And I'll come back again to these studies were run with incredibly tight monitoring compliance. And I've been involved in a lot of these studies. I've been on a lot of data monitoring boards where vitamin D levels of thousands of units were given to patients on various things of pregnancy. And I will tell you that we are still looking for our first adverse event due to vitamin D supplementation. On thousands of patients, we have not seen a single one. And so the paranoia about vitamin D supplementation at these levels in adverse events simply doesn't exist. And I would say that it's time for these monitoring boards just to not function anymore, because when you're looking at trying to find an adverse event that doesn't, it's a total waste of money. So I'd like to thank everybody for listening to this. I know it was quite involved, and I'd like to answer any questions that you might have. Thank you.
Thank you, Dr. Bruce Hollis, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your questions into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is vitamin D harmful during the prenatal or perinatal period?
It is not harmful, as I showed in our introductory material. And the societies have to get by this, like the obstetrics and gynecology. And the one thing I didn't mention in my presentation is, now here where I am at the medical university, where these studies have largely been performed over several years now, it was found that this decision was finally made here that all pregnant women coming into the practice now all get 4,000 units a day, all of them. So the faculty and the fellows and the residents have all been instructed that all women get 4,000 units of vitamin D a day. I guess finally the obstetrics department at our own university has seen enough data to suggest that yes, it is very beneficial and there is no harm in following this practice, and I'm quite proud of our university and the obstetrics here for instituting that practice.
Our next question is, why are your recommendations so vastly different than those currently recommended by many professional societies?
Okay. Okay. Again, I'll come back to the fact that the recommending societies are way backwards, and and very paranoid about making recommendations on vitamin D if it's not based on skeletal integrity. And you can see from the data that we presented that it's far beyond that. And to prevent asthma and preeclampsia and other things that wasn't the focus of this talk, like prevention of cancer and the prevention of autoimmune disease, go way beyond what it would be for skeletal integrity. But until the societies recognize that fact, not much from a standpoint of official recommendations is going to change. And it's up to the treating physicians and like this continuing medical education sessions for people to look at the data, synthesize it, and make up their own minds about how to best serve their patients. And that's basically the point I wanted to make there.
I would like to once again thank Dr. Bruce Hollis for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.